there's a coral reef. That coral reef is not an animal, like one animal or one plant. This is I understand, this is a collection of animals and plants and other and minerals are there. So those collection is not as a coral reef. So once I got somewhere like in a book that a Great Barrier Reef, which is around somewhere near Australia, uh, that produces eggs. So how can they produce eggs? And that happens only in the time in full moon in October or November. So that's what I was wondering.
I didn't get the second part of your question. Of course, I heard you're talking about the Great Barrier Reef in Australia, but what happens in the month of October? I missed that part.
Yeah, they produces eggs.
I
Many
see, I
corals, it
see.
is written like many corals produces eggs once in a year.
Once in a year.
I mean,
Okay, okay. So what is the question therefore?
so like this is not an organism. So how can they produce eggs? This is a collection of many.
I mean, it is only when you talk about laying eggs, it is only applicable to, let us say, fish. All right, because eventually you will see that in a coral area, you have certainly polyps, which are very small animals, which stick to the solids or to that calcium carbonate, which photosynthetic bacteria,
Ma'am, your
and
voice is
within that
breaking.
you will see fish are also. Oh. So within that area, you will see that fish are also swimming there. Okay, so when you talk about laying eggs, it is something which is only applicable to fish in that area. So there,
So
you have to use your discretion that this particular part, it only refers to a set of biological systems which are capable of laying eggs. Because eventually fish also becomes a part of that system. Okay, so let's move on now. And there is, of course, one lecture which I have missed, which is on emerging pollutants. So we will come back and we will try to see how we can make up for that. Okay, so let me get started with is the first slide visible here, which says lecture 6 and 7, microbial ecology and applications. So can you see this slide?
yes.
Students, other than Ratnesh, are you also attentive?
Yes, ma'am.
Yeah, anybody else? Okay, fine. Okay, so let's. You were saying something?
No, ma'am. No, ma'am.
Okay, so see, today what I will be talking about, and which is also a part of your course structure, is a little bit about microbial ecology and its applications. So in the last few lectures, we had discussed about ecology as a science of the environment and the interactions and the science of the interactions which are occurring between different components in an ecosystem. However, when you use a term like microbial ecology, you are going to scale down. When you are going to look at small microbes and how they are going to be interacting with each other. Okay, so it is just that everything is now going to be scaled down to be looking at what is called as microbial community, and a study of those microbial community means it is microbial ecology and what is its applications. Now, to begin with, let me first explain as to how microbial communities can be defined. So if you see, like in this particular picture on the left, we have the terms which we have already learned, and that comes from the subject of macroecology. All right, and this was when we discussed about ecosystems and communities, etc. We used many of terms. So starting with, for example, a very, very small level, which are the biomolecules. We talked about how when you talk about biological organization, you move from macromolecules to cell. A lot of cells will get together to give a rise for example, the cardiac cells, the heart cells will all get together and they will give rise to the heart tissue. Okay. And then these tissues, for tissue is nothing but a cell mass, but then they can become organized and give rise to an organ. Organ moves to organism, then you have large number of the same organisms which are called as population. So these are the terms which you learned when you were talking about ecosystems. Now let's move to how these terms can be understood when you talk about microbial communities. So here also we begin with macromolecules, biological macromolecules, and then you have a small cell. Now a small cell can be plated out on an agar plate, for example, which are going to multiply eventually, and it gives to a colony. So you see something like, which appears physically, which is undifferentiated, but they remain all together and they become visible in the form of an colony on an agar plate. And then, you have, for example, it starts to grow and, grow and assume a very, very big size, and it becomes what is called as a microcolony. However, what you would see in a single macroorganism. For example, a rat is composed of these kind of different organs. And here you will have now all these colonies, etc. You have now out here. So in essence, it is very similar to how you would describe in the ecological term as an organism. Okay. Next, we move to a microbial population. Okay. That is, these cells are now the clusters of these cells are going to come to environment, and then they make up what is called as a microbial population in as many ways as, for example, you will see that in this particular population, you have all these rats which are coming together to make a population. Okay, so a microbial population, therefore, is something what you see here when different populations come together. So different cell clusters, they come together, which could be very, very diverse from one another. Then you see something like a microbial community come up. Okay, so in other words, you have to work your way backwards to understand that microbial community is nothing but a collection of cluster of cells, and each one of them could be very, very different. There could be two or there could be multiple collections of cells which have come together in an environment because they are linked together by a similar function that they are going to perform. Okay, take the example of a wastewater plant. Okay, now in a wastewater plant, what you will see is that there are different groups of these microbial cells come together. One population could be something which is going to break down into individual monomers, like proteins can be hydrolyzed into amino acids, etc. Carbohydrate polymers can be broken down into sugars, etc. Then there is another set of population of microbes which is going to convert these sugars, etc., into volatile fatty acids. Okay, so in other words, now you have a second population of cells which is carrying out a different function, but nevertheless it is of an overall process, which is the anaerobic digestion process. So when you have these different cluster of cells that are now united together by the same function, okay, then gather together to carry out a similar function. Is that term clear to everybody as to what do we mean by a microbial community and what is its analog in the normal ecosystem which we had described earlier? Okay, so at the macro level in ecology, we use the term like community, where you could have a tree, you could have fish, you could have animals, etc., which are spread different type of, these are phytoplanktons, these could be some other population, this could be something else which is present in a small lake, for example. So this microbial community is united together by the kind of a function that they carry out. All right. So is this concept clear to everybody? that what do we mean by a microbial community? All right, very quickly, yes or no? All right. So now let me just read out. Let me just read out these the material which is written in the boxes. That this microbial community now, which is composed of different microbial populations, could be occupying. It could be in the lake, or it could be in the soil, or it could be in a wastewater treatment plant. All right. Now, microbial populations could be very, very diverse. So you could have hundreds of different these populations which have come together, and they are all united together to carry out a function which is going to be a single function. They could be taxonomically very homogeneous. That is, you may not see too much of a variation there, or it could be mixed, but nevertheless, they are going to be combined by identical physiological function. So all of these microbial populations have come together to carry out denitrification process, or nitrification, or photosynthetic process, or sulfate reduction, etc. So these microbial populations, in other words, have all come together in a community form in order to carry out some kind of an identical physiological function. Okay, and that is what we want to know, that what kind of communities are we talking about, and is there a way for us to determine, what kind of microbes are actually present within these communities? And not only do we want to identify them, but can we track them in a process? For example, in a way, we know that the microbial communities have come together in order to perform the final function of removing all the organic matter. So the COD, the BOD levels are going to be brought down by the microbial action. And you may want to know, as a function of time, what kind of microbes are really existing in there? How do they change as a function of time? Because as the organic matter, let's say in the sludge or in the wastewaters, as the organic substrate decomposes with time because of microbial action, you want to know, is there a change in the microbial community? Which microbial populations are going to be more effective, which are not so effective? And do I have a way to monitor these, etc.? Because eventually, a tool in order to monitor a process. Okay, and monitor also means that you not only can you study them, you can monitor them, and you can also control it at some point of time. So there could be a wastewater treatment plant, which you are trying to run, and by virtue of your ability to monitor the microbial community composition, you are also keeping an eye on the health of that particular reactor. If the community changes in there, you know that my reactor is not operate best, because under best conditions where my methane production, etc., is ideal, then this is my microbial community. But under altered conditions, my microbial community is not what I'm looking for. So it can also serve as an indicator of the functioning of a particular plant. So is that clear to everybody as to what we mean by that? Okay, so very quickly, I want to reply to this. So do you understand that why is it important to understand about microbial community? And, you want to determine how communities can be monitored. You want to know exactly what it is made up of, what mechanisms are there, how does it change as a function of time. In a wastewater treatment plant, the process goes all the way from degradation of the solid matter to formation of methane. So you want to monitor these communities, and you would like to use them, for example, as an indicator of the activity. So for all of these purposes, it's very necessary to monitor them. And of course, then you want to know, how do these communities, they respond to abiotic factors? And suddenly, your reactor is operating outside, and you find that the temperature has gone, the normal temperature would be 37 degrees, but then now it has become 43 or 44 degrees. So how does temperature, or how does pH, for example, alter the composition of the microbial community? How do they respond? So that eventually, the reactor operations, whatever I'm going to run, my process I can control, and my process can be very effective. So now as it says like down below, that when you look at this community, okay, it could be as simple as only two populations, because remember, community is at a higher level of organization. Below this, so a microbial community could be composed of only two populations. That is, it could be very, very simple, such as, for example, there are microbes which are sitting on handrails. When you touch, when you go up the stairs, you are touching all these handrails, etc. And they are very rich sources of these microbes, bad and good. Or doorknobs, for example, or light switches, or mobile phones, or clothing, etc. Okay, there you will see not too many populations. It could be very simple on these sources. Okay, or it could be slightly complex, such as you could have cyanobacteria, which are actually blue-green algae, or sulfate-reducing bacteria and other small microorganisms in a stratified means where they are layered microbial mats. So it could be just maybe 3 or 4 different, or it could be extremely complex, as you will see in the case of soil, where thousands of species per gram of soil would be present. Okay, or these could be very, very specialized communities which are going to operate in rhizosphere. What is a rhizosphere? It is actually the soil which is surrounding only the roots. So that particular area of the soil is called as rhizosphere, where the roots are making an intimate contact with the soil. And there is a very diverse microbial community which is operating there. And these are called as specialized communities because, there are only just as many things you can find when between the soil and the roots. So it is only to describe and to give you an idea that when you are going to use a term like microbial community, it could be very simple like it stays here, or it could be slightly complex, or it could be very, very complex. By complex, what we mean is that diversity. There could be thousands of bacteria which are going to be there. Okay, so that's what we're trying to say here. So now our question is that have understood the microbial community. Yeah.
Ma'am, I wanted to know what kind of microbial indicators are used in a wastewater treatment plant?
Certainly, I will come to that. I will come to that because you see, in the wastewater treatment plant, there are different kind of activities going on, as you know. So the first part is hydrolysis, which is breaking down of the food material into individual proteins will get broken, carbohydrate polymers will get broken down into sugars, starch will get broken down into sugars, etc. Then the second process is production of volatile fatty acids, which is called as acidogenesis. Okay, now second phase is acidogenesis, third phase is aceto, which is acetic acid production, fourth phase is methanogenic phase. So for each one of these phases, there is always an indicator bacterium which is going to be used. Okay, so for methane formation, there is an indicator bacteria, and that methanosarcina or whatever, which is, it makes a special cytochrome P445, which is responsible for methane formation. So only the genes which are going to be coding for that particular protein, which is the cytochrome P455, will be used as a set of primers in order to amplify out the 16S ribosomal RNA of methanosarcina, which will be an indicator bacterium that yes, my methanogenic activity is going on. Similarly, for hydrolysis, there will be an indicator bacterium. Is that clear? So for these different 4 phases which are operating in a typical, there are going to be indicator bacteria. So one enzyme which is very, very unique to that particular bacterium of that phase is going to be utilized in order to monitor what happens to that particular population as a function of time. So now, having understood now the microbial community, again, please, try to understand very, very diverse the communities are, and we are basically trying to link it to a process, which could be wastewater treatment, or it could be soil bioremediation contaminated with petroleum fuel, and a community of microbes, which is going to degrade all of this hydro completely into carbon dioxide and water. That means you are now bioremediating that because the community. All right. So now, having understood what is the significance of these microbial communities, and incidentally, whenever you try your bioremediation process, you will see that like getting rid of these hydrocarbon pollutants in the soil, it is never a single microbe which is capable of doing that. It will always be a collection of microbes which will be capable of carrying out that process. We have understood, and that's why the focus has shifted to studying communities now, not to individual microbes. We have already learned so much. We know about biochemistry of individual microbes, but since we know that we are going to be using only communities in the long run, in order to develop a biotechnological solution to a problem, we must understand the concept of communities and how do they respond. So our questions are, how do these communities, they respond to change in the environment, which is essentially the abiotic or the non-living factors. Okay, so if the nutrients become microbial communities, if the pH changes suddenly, if the temperatures change, if the substrate availability changes, do they all change at the same time? Or does individual population respond the same way as every other population? What governs? How are they going to respond? So I have a reactor now, a wastewater treatment plant, and suddenly I find that the incoming effluent in there is highly alkaline. Okay, it passes through an area where it has picked up alkali, etc. So now it enters into the wastewater treatment plant. Now, how is it going to impact my first part, my hydrolysis part? Now, in that part, I do have communities, but are all the individual populations in that community going to respond the same way? Is there a law for them? Do they respond differently? So that if I want to model that system tomorrow, I can do that. There are going to be growth rates which are going to be affected. There are going to be death rates which are going to be affected differently for each one of these microbial population. Okay, so it allows me to make a system more predictable. So the answer to that question is that certain principles which are drawn from ecology can be applied to answer that question. So a change in the abiotic factor, how is it going to affect my microbial population? Okay, so yes, I can answer to some extent with the question is, how do these microbial populations, they interact with each other? Okay, are they going, my question is going to be, if I have 10 different microbes which are in the hydrolysis part, are they going to interfere with each other? To what extent? Or are they going to be helping each other? So I want to know, how do they interact with each other, and do they remain the same as a function of time, and what decides the changes? Examples. So answer to that question is that examples seen in the environment can be used to answer that question. And we will take various examples to see. I'll do a case study with you to show that the microbial community dynamics, how they can be monitored in a wastewater treatment plant. When we have understood about microbial communities is that how do they respond to the biotic factors? Okay. Now, in the first question, we were asking,
Ma'am, your voice is breaking.
Yes, ma'am. It is breaking.
the presence of other microbes.
Regular intervals it is breaking. Regular intervals.
So the internet is back here. So should we resume now?
Yes, ma'am, we can.
I'm sorry, but you know, I guess the 10-minute break which we used to take is already taken in there. So let me just get started with the PPT. And the recording is already going on, so please give me a minute. Okay, so let me know if the slide is visible now. Slide number 4.
Yes, ma'am, yes.
Okay. Sorry for the interruption. So now, you know, getting back to like any ecological principles which can be applied in order to predict how the microbial population is going to respond. Now, one of the laws which is used in ecology to explain that is called as the Liebig's law of the minimum. Okay. Now, what it indicates in this slide is that if you look at any population, for example, which you know, a given set of microbes, there is always going to be one particular factor which is going to be the determining factor, and which will allow that microbe either to grow or not to grow. Okay, and that particular factor is called as the minimal factor, and the law is called as law of the minimum. In order to illustrate that point, you look at this particular barrel, okay, and you can see that there is, you know, all these pieces of wood which you see are called as staves of the barrel. So right here, what you are seeing is that there is water in here, and when you try to compare it to microbial growth, each one of the staves actually represents the different kind of nutrients which are required by the microorganisms. So there could be magnesium, there could be calcium, there could be nitrogen, phosphorus. They are required in different quantities. However, there is one particular nutrient which is required at a certain level, and if you were to fall below that level, then you will see that the water will start to fall out from this barrel. Okay, so is the analogy clear to everybody? Just as, for example, the retention of water in this particular barrel is dependent on what is the length of the potassium stave here. In a similar manner, for the microbial growth to occur, there is always one particular nutrient which is going to be the rate limiting nutrient, in other words. And that was defined as the Liebig's law of the minimum. So I hope this analogy is clear to everybody, that in that, therefore, you know, you don't have to worry about 10 factors which are going to affect the microbial population at a given time, but it could be just one particular factor, and that is the minimal factor, or the law was therefore called as the law of the minimum. So, for example, crop plants. Now you will see that the growth of the crop plants is limited by the most limiting mineral nutrient. Okay, and so mu refers to the specific growth rate, is directly proportional to the concentration, which is the you know, S1 and K1, etc., is the first order constant for the utilization of this minimalistic factor. In other words, the growth rate is going to be regulated by the rate at which that particular nutrient is going to be utilized. Okay, so that is called as the law of the minimum. So, like example, crop plants are going to be affected by the availability of nitrogen. Similarly, the phytoplanktons in the ocean, that is, these are photosynthetic microbes which are in the ocean, are going to be controlled by the availability of iron. Other factors, whether it is dissolved carbon dioxide, whether it is nitrogen, potassium, etc., are not really the limiting factor, but availability of iron is the limiting factor. So the growth rate, so it is very much like, you know, the Monod's model, which you have learned, that you can always relate the growth. If you look at the growth rate equation, then the growth rate is going to be controlled by the availability of a particular substrate. Okay, so the same as Michaelis-Menten type of a kinetics here. Of course, the substrate refers to the level of a particular nutrient in the medium. So that's what we mean here. So phytoplanktons in the ocean, therefore, are going to be controlled by the availability of iron. Heterotrophic bacteria, yeah, there is a question.
What is it is minimum?
Minimum. It is the concentration of that particular nutrient which is available in the minimum quantity. And the growth rate is therefore going to be determined by the availability and by the utilization of S1. K1 is the rate constant which you are using for the nutrient which is Si. Okay, so is this clear?
Yes, ma'am.
Okay, and similarly,
Can you please explain again what is Ki?
What is Ki? Ki, like it says here, I have written it down, is the first order kinetic constant. It is the rate at which that particular substrate is going to be utilized. Availability could be there, but there is a certain rate at which it is going to be taken up by the microorganism. So that rate constant is the kinetic constant for that. So in ecology, therefore, so when you talk about a microbial community, again, extrapolating this to a microbial community, we can say therefore that, in a microbial community, the populations are going to be restricted by the availability of only one nutrient, not by 10 other nutrients which are going to be affected. So this was called as the law of minimum. However, the limitation of the Liebig's law. Now, in the lab conditions or in a chemostat, because, eventually people started to say, well, this is a very generalized kind of a term, but if I try to grow my microorganism in a chemostat under laboratory condition, I find that this really doesn't apply. Okay, so the organism's growth was shown to be affected by several nutrients at the same time, and Liebig's law was said to be a simplification of the whole thing. In fact, there are interactions between several nutrients which could also be occurring. Now, communities are not stable, but they are always moving from one steady state to another. The effect of external factors and metabolic factors can change that. Okay, so it would be very hard to say that the microbial communities are, going to be constant as a function of time, and therefore they are going to be dictated by the availability of a single nutrient. No, the communities are also changing it's a very, very dynamic community. It is changing with respect to time. Why is it changing? Well, because the availability of nutrients, the supply of nutrients also keeps changing. So the communities are also going to be very different after 10 days. They are going to be very different. So therefore, it is just to say that this particular nutrient is going to be limiting. See, because eventually what you want to do is that once you identify this law of the minimum, in other words, you are also able to identify the limiting nutrient. So if you feel that, well, nitrates are going to be limiting in my reactor, then is it enough for me to supplement that reactor with nitrate and ensure that the microbial communities are quite healthy there, and they will be performing the desired function of, let us say, methane production? That is the reason why I'm trying to understand what are the implications of law of the minimum. So if it is really true that they are dependent on only one nutrient is going to limit, so what if I add after 10 days, if I just add a little pulse of nitrate, will my community dynamics be all right and will everything be restored? Okay, so that is where we are trying to extrapolate and we're trying to learn. Now, the second law is, so under ecology, then the law of tolerance became available. So which is called as the Shelford's law of tolerance, which indicated that every system, every microbial population, for example, it has a certain optimum range, like is shown in this particular figure. Okay, and this optimum range could be either pH or it could be temperature. However, there is the law of tolerance. That is, there are zones in which they are able to tolerate. So if the optimum temperature is like, maybe 35 to 37 degrees, but it can even go down to 27 degrees. On the other side, it can go down to maybe 40 degrees also. So in other words, there is no hard and fast rule. And when the law of the minimum was applied, that is the first law, Liebig's law of the minimum, it was applied to, for example, to nutrient conditions. But whereas the law of tolerance was now applicable to abiotic factors, such as pH, such as temperature, etc., not to substrate concentrations. Okay, so the applicability of both the laws should also be understood in terms of where they are going to be applied.
First
So,
law is only applicable to, okay, ma'am.
yeah, so it just, in the next slide, I have just summarized these points that the Liebig's law, which is the first law of the minimum, is applicable to factors which are actually the resources. That is, the resources like a nutrient, or it is dissolved oxygen, or it is the water content in soil, for example. So the Liebig's law is applicable to such kind of factors, whereas factors which are the state of the environment, such as temperature, pH, electrical conductivity of the soil, etc., are considered within the tolerance law.
In the second law, what exactly are the implications if there is beyond a certain limit? We have the state of
Okay.
environment.
Okay. See, because the question is that how much, now, for example, you are operating a wastewater treatment plant. All right. Now, ideal condition would be 37 degrees, okay, for it to operate, where bacterial communities are operating. And, all these wastewater reactor plants, they operate outdoors. You don't keep them in an environment. You have the effluent which is collected, etc. So now what happens, you would like to know that if there is 45 degrees, can my reactor really operate? Do I need to shut down because the reactor is not going to operate? So the law of tolerance can be applied under those conditions, that no, look, I don't have to shut down my reactor, or I don't need a jacketed vessel in order to provide a constant temperature. But because of the law of tolerance, see, these are actually common sense, but you want to put them in scientific terms now. So for operating my wastewater treatment plant, I don't need a jacketed reactor where the temperature has to be always monitored or kept between 37 to 40 degrees. No, even if the temperature rises to 44 or 45, maybe the reactor will not be as efficient, but nevertheless it will operate. Or if the temperature goes down, slightly colder conditions, the temperature becomes like 30 degrees, can my reactor still operate, or do I need a jacketed vessel now in order to keep 37 degrees? So we need to understand this law of tolerance in terms of its applicability at that level. Because there is a tolerance towards temperature, because there is a tolerance towards pH of the microbial population, towards electrical conductivity. Maybe there are too many so you have now your effluent passes through an alkaline field, and the pH becomes very, very high. Conductivity is very different. Then do I need to set all those conditions before I actually take it into the reactor? So for those conditions, the tolerance law makes sense, that no, it can still operate within, because there is a zone in which it can be tolerated by the microbial population. So the implications are that I don't need to have a jacketed reactor. I don't need to spend money in order to control the temperature or the pH of the reactor. So does it make sense?
Yes, ma'am.
Okay.
So, ma'am, these two laws should be considered as complement to each other. So, ma'am, isn't it like one law? If even Levik's law we apply, we can have like Levik's law for temperature, like this much temperature after below this temperature, we cannot microbial population will not grow. Something like that. Can't it be like?
See, that's why we are not trying to do that, because in that slide it showed clearly that the Liebig's law of minimum is being applied to resources, which are substrates, which are dissolved oxygen, etc. But because for temperature and other things there is a law of tolerance, so they have been
Yes,
kept separate in the ecological field.
So understanding it easily, we have kept it.
Okay. Now, for this, let me go to full PPT mode because the font size is small. And if you have trouble viewing the slide, then let me know. I'll just put it on the full PPT mode. So can you see the slide now?
Yes, ma'am.
Yes, ma'am.
Okay, all right. So broadly. So now the thing which we are trying to ask is that in a community, there are different microbial populations. Okay, what kind of interactions are likely to occur among the populations? There are different kind of interactions which can occur, and all of these are listed here in ecological terms because it is important for you to know, that what exactly is happening between these microbial populations. Either you can have the first point says mutualism or symbiosis which is occurring. Now, what exactly is it? It is, now if I give you an example, it will be a lot more clear. It is the relationship of leguminous plants. Leguminous means your cereal, dal, etc. All these plants with the nitrogen fixing rhizobia, which are the nitrogen fixing bacteria which are present in the roots of these plants, okay, in which the plants are going to provide the bacteria with organic carbon substrates and a growth habitat in exchange for fixed nitrogen, because otherwise nitrogen has to be supplied from outside as a fertilizer. But then in the case of leguminous plants, there is a natural association of their roots with the nitrogen fixing bacteria. So these bacteria, they take up molecular nitrogen, they take up hydrogen from the environment, and they make ammonia. This ammonia can be used as a nitrogen source. Okay, so this kind of an interaction can now occur. What is this kind of an interaction called as? It is called as mutualism, in which both the populations are going, upward arrow means that both populations, they benefit from each other. Okay. Then there is synergism. That is another kind of interaction which can occur, in which again, both the populations are going to benefit from the presence of each other. But then it is different from mutualism. How is it different? Let me give you an example. In this syntrophic association, for example, between the fermentative bacteria and the methanogenic archaea, which are often present in the wastewater treatment plant, the bacteria are going to hydrolyze the organic compounds, ferment the products, and they produce hydrogen acetate and format. That is formic acid. These materials are then consumed by the methanogenic archaea, thus maintaining the concentration at low level and keeping the overall fermentation reactions energetically favorable. Okay, so in these cases, they are highly dependent on each other. You see, in this case, they are benefiting from each other, but they are not dependent on each other so much, because the plants can still get nitrogen from outside, and the nitrogen fixing bacteria in the soil, there are going to be nutrients available anyway. They can also get it, but the presence of plant is going to facilitate that. It's going to accelerate that process. Okay, in this case, they cannot survive without each other. So that is the difference between synergism and mutualism.
Just one doubt in mutualism.
Yeah.
Ma'am, you said that they take up hydrogen from the atmosphere and they have the nitrogen fixing bacteria, so they form ammonia. So ma'am, the simple question is, do they directly form ammonia with the help of N2 plus H2? Or this
Absolutely. No. Nitrogenase is the enzyme. We will have an example, later on in the lectures, that these are nitrogenase enzymes which are only present in the nitrogen fixing bacteria. Not all bacteria can fix nitrogen. So these are only in the root nodules of these leguminous plants, like you have arhar, for example. If you pull out that, it's a shrub, it grows to a relatively bigger size, but if you look at the roots, there are these nodules which are kind of like tumor. It looks like a fat tumor which is present on the nodes, on the roots, and within it is a population of these bacteria which take nitrogen from the atmosphere, which take hydrogen from the atmosphere. In the presence of the enzyme nitrogenase, they just convert it into ammonia directly. And these are highly, oxygen is toxic to them. mind you, that they are highly anaerobic bacteria. They carry out this process in the absence of molecular oxygen. So the nodes will, that bulbous portion will provide them with the anaerobic environment in which this particular reaction can be catalyzed. Okay, but the bacteria are not really completely dependent because organic matter could be available in the soil also. Same way, the nitrogen containing, if you are giving fertilizers and other things to these plants, you don't leave them at the mercy of the nitrogen fixing bacteria. If there is a field which is growing arhar dal, it will still have fertilizers. But the process is accelerated. You will have to give lesser fertilizers because there are nitrogen fixing bacteria which are already in there. And number two, sometimes, the soils, they also become drained out in these because of fertilizers and all that. Many of the microbial populations are killed. Then there is a crop rotation. So you bring in a leguminous plant between rice and wheat crop. For a few months, you're going to plant the leguminous plants because then they restore the health of the soil. Because ammonia is made, nitrogen again goes back into the soil. So this way, there is crop rotation which is being done in all of these. Okay, so anyway, so these are the two kind of interactions which are listed here. Then there is an interaction called as commensalism, in which one population, pretty neutral, it doesn't matter, but the second population gains. For example, you have cyclohexane, which is an aromatic pollutant coming out of petroleum products. It is contaminating the soil. So there is a microorganism which has the right enzyme to convert cyclohexane into cyclohexanol, which is by monooxygenases. Okay, so if you remember, like methanol, if you look at methanol from methane, so methane is CH4. So if you have a monooxygenase enzyme, it adds a single atom of oxygen. So now you have like CH4O, that is nothing but methanol. Okay, so same way, there is a microorganism which makes a monooxygenase, which can convert cyclohexane into cyclohexanol by one microbial population, and it gains no energy from the process. It is just that it keeps producing that enzyme. In the process, there is a cyclohexane sitting, it gets converted to cyclohexanol. But however, there is another second population which is able to carry out, which is able to metabolize cyclohexanol, in which case it benefits. And that's why we show that, okay, it can use it as a carbon source. So such kind of an interaction in the microbial populations is called as commensalism. Then we have neutralism, in which none of them, they really benefit from each other. For example, you could have two spatially separated microbial populations which have no interaction with each other. They don't get affected by each other's presence at all. That will be called as neutralism. Or there could be predation or parasitism, parasitism, like a parasite, in which case one population will go up and the other population will go down. So, that's pretty easy to understand. Predators like protozoa which graze on the bacterial populations, or there is amensalism where you have either neutral or upward and this goes down. Again, there is an example which is given here. Or there could be simply competition, that they fight for the same resources, and therefore each one of them, their population is going to go down. Okay, so it is just to give you an overview of the kind of, so when you think about a microbial community which is going to be operating to carry out a process which you would like it to carry out, as a technological intervention somewhere, you have to keep in mind that these are the kind of interactions which are going to be. And sometimes, we also create based on what we have learned from the natural, what is happening in the natural environment. I want to create a synthetic microbial community because I think, well, I have put all the ingredients together and now I can develop and I can just put it in a polythene bag and I can supply it to somebody that, look, this is going to do an excellent job for you. But then you have to know what kind of microbes to select, what kind of interactions are likely to occur. And in the field, they should be, they should be beneficial and they should not hinder with each other's action. So that is the context in which one must learn all this. So is this clear to everybody?
Ma'am, do
Ma'am,
you
just
think
one question.
communalism part, ma'am, what is the significance? What is the working of the first population? What is that first population doing?
Okay, the first for example, there is cyclohexane as a pollutant. There is a group of microorganisms, bacteria, which makes monooxygenases. So what it does is that it makes extracellular monooxygenase, which is secreted into the medium, which is secreted extracellularly, and it is capable of converting cyclohexane to cyclohexanol. That's an enzymatic reaction. So this particular microbial population, what it does, that it only converts cyclohexane to cyclohexanol, but it doesn't go beyond that step at all. It cannot do anything with that cyclohexanol. However, there is a second microbial population in that community which can take up cyclohexanol. There are, so many yeast, so many fungi which can take up these C1 compounds which have alcohol groups as a carbon source. These are methanotrophs. They grow on methanol compounds. So cyclohexanol can be taken up by this second population. It can be metabolized to carbon dioxide and water. It can be used as a carbon source. So eventually you have achieved your goal. Your goal was to convert a pollutant, which is a cyclohexane, into carbon dioxide and water. That means you want to do mineralization. You want to completely break down this hydrocarbon into carbon dioxide and water. That means mineralization. That also means removing this pollutant completely from the environment. But now you will require two populations. First, which is capable of carrying out, so it changes cyclohexane to cyclohexanol by addition of a single oxygen atom using the enzyme which is, these are all aromatic. So it could be an aromatic monooxygenase. There is methane monooxygenase, there are ethane monooxygenases, so on and so forth.
Yes, ma'am.
So is that clear as to what it is doing?
Yes, ma'am. Yes, ma'am.
Okay, okay. So
And ma'am,
let's, yeah.
just one thing, ma'am. The arrow downward is negative effect. Negative effect on the population which we are talking.
Negative effect on the second population.
Yes, ma'am. Understood.
Okay. And of course, these terms are defined. If you just see at the bottom of the table, all of these symbols are also explained here. Okay. So now, is the slide visible to you? Yeah.
Yeah. So, ma'am, instead of using microorganisms, what if we use directly the enzymes? Is it feasible to use enzymes or for that benefits which we are getting from microbes?
Then the question is, one has to weigh out the advantages of using whole cells vis-a-vis enzymes. Now, for certain things, like in the chemical industry, for certain processes, pure enzymes can be used. Okay, now supposing you want to convert, you have one particular metabolite, acetone has to be converted into an alcohol. Okay, so in the pharma industry, there is an acetone compound which has to be converted into an alcohol compound. If that specific reaction is to be carried out, then enzymes are preferred. Why? Because microorganisms are going to have hundreds of these enzymes. They will be non-specific, so on and so forth. Now, if that is the kind of specificity which you want, only then you will use enzymes, because you have to cultivate the organism on large scale, then you have to purify the enzyme. Enzymes are very, very labile to temperature. You will have to freeze dry them. You have to increase their shelf life, so on and so forth. So it's very, very expensive to use enzymes. Whole cells, on the other hand, the cells can be just grown in a bioreactor and you freeze dry them, you lyophilize the cell, put it in a package in a plastic bag and give it to the farmer. And you tell him that these particular microbes are capable of making a bioinsecticide. So what he does, he will just take that polythene from you and he will only spread it out in the field. So using whole cells is going to be a lot more easier. The shelf life of cells, they are not so finicky. They can be stored by usko up 30 degrees pe bhi rakh do, nothing is going to happen. They can tolerate the temperatures, etc. But enzymes are very, very finicky. So the storage conditions, the purification, and it's far more expensive to produce enzymes. So therefore, it is always preferred wherever you can carry out things with whole cells, people try to have the first preference as whole cells. But if you can't do that because of specificity or because of some other problems, then you deal with individual enzymes as biocatalysts. So is that clear?
Okay. Yes.
Okay. So now, I'm going to just discuss with you for example, the wastewater treatment plant. And I'm just going to again and again come back to this process because it is an excellent example of how different microbial communities which perform a function have come together in order to start with the nutrients which are present in the wastewater, and they completely remediate it, they bring down the BOD levels, etc. And they also produce energy in terms of methane can be produced in the process while the treatment. Okay, so first of all, in this anaerobic digestion process, there are 4 phases, as I had mentioned very briefly. The first phase, like you see here, okay, so can you see my my mouse here, or shall I go back to that other?
Yes,
Okay.
we can see.
Okay, I don't want to have those funny laser pointers and other things. So if this is visible, I would rather use this. So phase 1 is called as the hydrolysis phase, where complex biopolymers which are coming in the wastewater streams, such as proteins, polysaccharides, fats and oils, are converted into individual monomers. So from proteins, you are going to get amino acids. From polysaccharides, you are going to get sugars. And from proteins, of course, you can get peptides, etc. From fats and oils, you are going to get acids, so on and so forth. Okay, so this particular phase is called as the phase of hydrolysis, and it is carried out by the fermentative bacteria. So it is a population here which carries out this. In the second phase, you will see, which is shown in orange here, is called as acidogenesis, during which volatile fatty acids are produced. Okay, what are these acids? These are short chain volatile organic acids such as propionic acid, butyric acid, or formic acid, etc. And these fermentative bacteria are going to use sugars, etc. and don't convert them directly into carbon dioxide and water. But since everything is happening in the absence of oxygen under fermentative condition, which is lack of molecular oxygen, okay, so you have propionic acid and you have butyric acid, etc., which is produced. And so acids are being produced. Okay, some fermentative bacteria can also convert, for example, directly these sugars, etc., into hydrogen and carbon dioxide, or some fermentative bacteria can also convert these into acetic acid, whatever may be the case. But in the event that volatile fatty acids are produced, converting these into hydrogen, carbon dioxide, and acetate is called as an acidogenesis phase. That is, during this phase, acetic acid is produced. This is the third phase in the anaerobic digestion process. And the fourth phase, which is shown in blue, is called as methanogenesis, where either from acetate, you can convert acetate into, because you have to reduce acetate. Acetic acid is reduced, and then you can get methane and carbon dioxide. Or in the event that molecular hydrogen and carbon dioxide are there, there is another group of bacteria which can convert this now into methane. So this is called as the methanogenic phase. Okay, so now you see that very, very complex microbial populations are operating in a wastewater treatment plant. If you want to look at the yeah.
Ma'am, are the fermentative bacteria already present in the wastewater treatment plant?
Absolutely, absolutely. Like when you release them today, of course, you start out with you start out with sludge, for example. The sludge is already recycled back. That is in the activated sludge process. But that is only one way of treating the waste. The other way of treating is anaerobic digestion, under which conditions methane can be collected. Okay, so in this particular process, okay, there are bacteria, which are acclimatized because if you throw this into a water body, let us say the domestic wastes, etc., are released into a water body, into a nearby pond. Gradually, over a period of time, the bacteria are already in the atmosphere. You don't have to add them from outside. They start to seed and they will directly start to treat. You see what is happening in this, in these, you see these landfills, etc. There are fires which are happening. ये जो आपके गाजीपुर का landfill है, तिमारपुर का जो landfills है, you will often see that there is fire which is occurring at several locations. That is because there is in that landfill area, all anaerobic conditions have been created. There is no molecular oxygen. And all these bacteria have naturally come up in those populations because they are everywhere in the atmosphere. Okay, but what we do, like if you want to now have some kind of a control, then this stream is going to be released. You know, supposing you want to set up a plant and you say that, okay, for a community, for a locality, let's say Vasant Kunj locality, all this waste, domestic wastes are going to be collected, and then I'm going to set up a small reactor, and we will now, we don't want to waste it. We will treat it certainly, but the sludge which is left behind, I'm going to convert it into methane, and I'm going to generate energy out of this waste. Okay, now in there, having the biochemical knowledge already, you may want to start with your own inoculum, which has been collected from a natural source. So you go there and you collect 10 liters of that inoculum and you use it as a seed to inoculate your own reactor. Okay, but it is not something which is synthetically created. It is something which has been taken from a naturally occurring consortium. So is that clear?
Yes, ma'am.
Okay, now if you want to examine the microbiology of the process, you will see it is extremely complex, very delicate, involving several groups of bacteria. You know, you have bacteria which can carry out this, you have bacteria which can carry out only the hydrolysis step, and then you have the acetogenic bacteria, acetoclastic, these are archaea, etc. So on and so forth. Each of these have their own optimum working conditions. These groups are sensitive to and possibly inhibited by several process parameters such as pH, alkalinity, concentrations of free ammonia, hydrogen, sodium, potassium, so on and so forth. So it gives you an overview of the complexity of these microbial communities in a naturally occurring process. Now, so microbial community interaction for an effective process control. Now you want to control, you know, you have set up a reactor, you have inoculated and things like that. Now you want to operate that reactor and you want to control it so that, you know, the ideal conditions can always be maintained under which maximum methane can be produced. For an effective process control, detailed knowledge about the microbial catalysts, that is, what kind of microbes are there, and their correlation to process conditions is very, very crucial. And their relationship to process outcome is also important. That is, if the process outcome is methane generation, then you want to know how each one of these microbial catalysts is really related to the final production of methane. Are all of them important? Or only some of them are important during this stage. So if my reactor is not operating very good, let's say in the methanogenic phase, can I just have a microbial consortium, which I know is correlated to this process outcome? At that point, I only add a selected group of microbes, and yet my reactor operations are going to be successful. Okay, so that's the reason why we want to say that the relationship of the microbial catalysts is very important to process conditions, but it's also important to process outcome. Okay. So here is an example which I'm going to give you of you know, how a microbial community can be monitored in a typical anaerobic digestion reactor. And so is this slide visible to everybody?
Yes, ma'am.
All right. So this is from a paper, and I'll also, I will also share. The title of the paper is Metabolic and Microbial Community Dynamics During Anaerobic Digestion of Maize Silage in a Two-Phase Process. So essentially what these investigators are trying to do is that they are trying to monitor the microbial community dynamics. How does it change during the anaerobic digestion process of maize. Maize is corn, silage, because after corn is taken away from the plant, then you have all these their stems are there, their leaves are there, which are cut out. They can be fed as a forage, as a nutrient, or it can also be taken and it can be dried, and then it can be used to generate methane. Okay, it can be used as an energy source. So in this, the investigators are trying to follow that if I were to set up an anaerobic digestion of this maize silage, then what are the metabolic activities which are going on as a function of time, and how does the microbial community change as a function of time while we go from degradation of all of these polymeric materials in the maize silage to methane formation. So it's in a two-phase process. What are the two phases which they have used in this? The first phase is, yeah.
I'm sorry for interrupting, ma'am. The maize silage, we have put it in the reactor and then we are taking the
Yes,
outcome.
absolutely, absolutely. Okay, so now your starting material is going to be the maize silage. Okay, that is your stems of the corn tree. Once the corns are removed, then you see that it's like very much like your sugarcane plant. It has a tall stem, not very strong, and then it has all of these leaves. Now they are dried and they can be now cut into, they can be chopped into smaller pieces, and they are going to be put into the bioreactor. Because essentially, what do the leaves contain? They are going to contain a lot of starch. They have chlorophyll. They have other polymers. Okay, there are membranes of those cells, etc. There are proteins which are present in the leaf and in the stem. So basically, these are all biopolymers which are present in the silage. Is that clear?
Yes, ma'am. It is clear.
Okay. So therefore, in the first, so the process says that this process is carried out in two phases. So the first phase is something like it explains here. In the first phase, you have the hydrolysis which is being carried out, and and acidogenesis phase are being carried out. But the kind of reactor system which they have used is a leach bed reactor system, and I will explain as to what it means. Okay, the second, so out of the four phases which I explained in the anaerobic digestion process, it is only the first two, the hydrolysis and the acidogenesis, which is being performed in this particular part of the reactor. In the second phase, which you will see here in this particular vessel, which is the second phase reactor, you have acetogenesis and methanogenesis, which are being carried out in this part of the reactor. So the entire process is being carried out in two phases. Now, the first phase is in what is called as a leach bed reactor system. In a leach bed, what we have, for example, these are four parallel columns which are set, and the solids are kept at the bottom of this column. And then there is a liquid which is pumped right here. Okay, and it is like, it could be acid, it could be alkali, etc., which is now treated, okay, gradually, so that sugars and other things are extracted and the percolate. Okay, so obviously there are filters which are going to be here, so that after acid treatment of all of these leaves and stems, etc., the sugars are going to be extracted, and now they will come into the percolate vessel. Okay, so exactly the same situation happens. So as soon as the first reactor is, emptied out, then the second reactor, the content of the second reactor, the percolate is something which just trickles down, okay, through a tube, and now it can be fed into this. So the idea here is that the reactor operation can be kept constant, because you go from reactor 1, then to 2, then to 3, then to 4, and then you again switch back to reactor 1, in which the process of treatment has already occurred. Okay, you have extracted, and now hydrolysis. So there are inoculum, the bacteria, etc., are going to be inoculated now already in this. So they are going to carry out hydrolysis as well as acidogenesis system. And now from the first reactor, what you will go is to the second reactor here. You see there is a pump which can either, it can be directly fed into the second reactor where acetogenesis or metherogenesis is going to be carried out, or for example, it can be cycled back and it can be stored in the intermediate vessel, so on and so forth. And at the same time, the gas formation. So there is a, because there is some kind of an activity which is going on, carbon dioxide, hydrogen keeps on getting produced. So you have a gas counter where this gas is going to be monitored. Similarly, the gas is going to be monitored. So periodically, samples can be removed from this. As the process operations go on, samples can be removed and you can see what happens to the microbial population, what happens to the various acids, lactic acid, propionic acid, etc., and what kind of gases are being formed. And that is how the dynamics of this reactor can be monitored. Is this clear to everybody?
Ma'am, the milligas counter, yes, ma'am. What is this, ma'am?
Well, in the sense that I don't know why they use the term milligas counter. Is it that even very small amounts of gases which are emanating from these processes can be monitored here? How much of gas is coming here? Yes, and similarly, how much of gas is being produced can be monitored through this monitors. Okay, so is this clear?
Yes, ma'am, it is clear, but like in this.
Yeah, please continue.
So,
one of you had a question. Yeah.
Ma'am, the hydrolysis gas, what is its significance? Like, can we use it as well?
Well, right now, okay, they are only being used, the gases are being used for monitoring purpose. That is how much of methane and how much of hydrogen and how much of carbon dioxide is being produced. But in the long run, methane can be, the whole idea is that this is of course an academic exercise that you want to know how much of methane you are generating. But tomorrow, if you really set up a plant and you are making a methane, there is no reason why methane cannot come back and serve as a source of energy to operate the same partially. A part of the biogas can be collected and it can be diversified. But a part of the biogas, can also be brought back into the system and it can be used to fuel the reactor to maintain a certain temperature or whatever. There is no reason why it can't be used for that also. Okay, but in this particular paper, it is only being monitored because you want to know the efficiency of the process which you have set up. So you have set up a two-phase process for digestion of organic biopolymers in the maize silage, and you want to make energy out of it. So you want to know how effective is my system? Am I making good enough methane, or is the methane yield so low? Is it a successful process or not? So right now, it's just an academic exercise. So is that point clear to everybody?
Yes, ma'am.
Yes, ma'am.
I think let me stop. Yeah. Yeah, please
so
continue.
Are they trying to like their focus is to understand microbial dynamics, or their focus is mainly on maize, so that if they want to do something, produce methane using maize, so they can enhance that process? Yeah, in this
absolutely
paper.
So right now it is just an academic exercise. It's a research paper where they have tried to do things a little bit differently. Normally, you have in an anaerobic digestion plant, all these four phases, they go on in the same reactor. But then what happens is that the products of the first phase or the products of the second phase could be interfering with the processes which are happening in phase 3 and phase 4. So that is the reason why they said, well, let's try your acidogenesis and acetogenesis phases. Let's try to separate and see if the efficiency of the overall process can be increased. Okay, so is that clear?
Another
What is
doubt. Yeah. Ma'am, the doubt is regarding that equation mu is equal to minimum KISI. Can you go to that?
okay? Okay. How are you placed? Because is there a class for you after this?
Ma'am, for me, not. There should be, but not.
I see. Yeah, please. So what is the question?
Ma'am, that mu is equal to minimum KISI when we are considering. So, like, what is mu? Like,
It is that is the
K
minimum.
is a specific. Mu is a specific growth rate, something like that. We consider. So K is also is
Yeah,
a specific
absolutely.
growth. Okay.
K is actually the first order kinetic constant of utilization of the substrate I.
No, ma'am, so mu is considered as like dx by dt into 1 by x. That is called a specific growth rate. And when you are saying that first order kinetic constant, so is it that k is equal to 1 only dx by dt or ds by dt in this case?
No, not at all. Not at all. In the broader term, it will not change. It is dx by dt into 1 over t. 1 over x, sorry. X dx by dt and
Okay.
1 over x. Okay. But the idea here is to show that the specific growth rate, okay, is going to be controlled. Because this is only a constant here, you know, is a kinetic constant. So it is the concentration. K is the constant here. Si is the concentration of that nutrient which is present in the minimum quantity. So out of many substrates, to S3, there would be only one such nutrient, the concentration of which is going to control the growth rate.
Oh, yes,
that's what it means.
Ma'am, can I leave? I have a class.
Okay, absolutely. I'm just going to stop right here and then we will come back and we will. So the lecture is over. I know you must be having a class now elsewhere.
If you have,
So,
I have a question. If you have